Okay, it is, we're recording now. Okay, hello everybody and welcome to CureGRIN's first webinar. I'm Denise Renner and I'm the founder and president. Can you guys still hear me okay?
Yep.
Okay.
Yep.
So I'm the mom of a child with GRIN1 mutation, and today we'll hear from Keith McArthur, who is the medical outreach coordinator for CureGRIN, and Keith is also the father of a child with GRIN1 mutation. We're joined by Eriskay Liston, genetics counselor for Hospital for Sick Children in Toronto. And today we're going to discuss genetics for GRIN families and reading genetics reports. I've already muted most everybody, but we're welcoming questions throughout the webinar. So if you have questions, you can unmute yourself, or you can also use the chat function at the bottom of your screen. And please note we're recording this presentation and we'll be posting it to YouTube. So keep that in mind if you're sharing any personal information. Okay. You want to, next slide. And here's our presenters. So CureGRIN is one of several proud sponsors of the CFIRB Conference on GRIN Variants in Atlanta, September 13th and 14th. And this conference brings together leading researchers from across the globe to share information and collaborate. So if you haven't signed up yet, I encourage you to do so. This webinar aims to help inform parents and build the knowledge base in order to prepare us for the information that will be presented at the CFIRB conference. Next slide. CureGRIN is committed to improving the lives of people living with GRIN disorders, and we work closely with scientists in the medical community to drive research that will lead to therapies and a cure. That said, there's a lot of work to be done, and we need your help. We're currently looking for other board members and seeking multiple GRIN types, and there's many other ways to get involved, such as helping with marketing efforts, fundraising, legal areas. If you are interested, please reach out to either one of us. So with that, Keith, I will turn it over to you.
Okay. Well, thanks everybody for taking time out of your day or night, if you're in Europe, to spend time with us. I'm really happy to be joined by Eriskay, and this is basically what we're going to go through today. So I'm going to kick it off with part one. I'm going to give kind of an overview of genetics as they apply to the GRIN1 genes and the GLUN proteins that those GRIN, all GRIN genes and the GLUN proteins that those GRIN genes create. Now, I want to say, first of all, that I'm not an expert in this stuff. I'm someone who only took biology up to grade 10, and I struggled in it. But since Bryson got his diagnosis, I've really been trying to learn as much as I can about it, and so wanted to kind of put together what I've learned and share it today. And I'm hoping that if I get anything wrong, or if you have any questions that go beyond my expertise, that Eriskay will be able to answer those. Then we're going to move on to part two, where Eriskay is going to take us through how to read and understand a genetics report. In part three, I'm going to talk about understanding your functional analysis. So this is specifically if you've had a functional analysis done at CFIRB in Emory. I was there a couple of weeks ago and was able to tour the labs and got some understanding from Steve Trainalis and the other researchers there about how they do that research and how to understand the test. So I'll take you through what I know, and then we're going to finish up by taking all of your questions. So I want to start this first section with some very basic research. I know that there's some people who, or some very basic science. I know that there's some people who probably have more knowledge than others, but I want to really start at the beginning because I know that there's, some people who have said that they really have no understanding at all. So I want to start as basically as we can, and apologies to those who, understand some of this already. But basically, what you can see here on the right hand of the slide is, a stylized version of the double helix that we recognize as DNA. And basically what DNA is a code that exists in every human, in fact, in every living thing has DNA, which is basically the instructions of how to create itself, how to build a human or an animal or a plant or a virus. It's those instructions and on how to build, and then how to kind of repair and maintain the organism as it grows in life. And so what is DNA? DNA is a language. So just like, you can see on the left, there's a bunch of different languages. Our spoken languages. In the middle, you can see the language of music, and on the right, you can see DNA. And every kind of language has its own sort of alphabet or code. So with spoken languages in the Western world, it's the traditional alphabet that we know. With music, the language and the code is notes and bars. And with DNA, the language is actually communicated through 4 tiny particles of chemicals that exist in the body that we know of as, or scientists, researchers refer to as A, C, G, and T. And we don't need to get into details about what those chemicals are, but just suffice to know that the DNA is a long sequence of these chemicals that are represented as A, C, G, and T. So just how long is the sequence of DNA? Well, War and Peace, which is considered one of the longest novels ever written, it has approximately 3 million letters of the alphabet in that book. Well, human DNA has approximately 3.2 billion letters. So it would take 1,000 War and Peaces to contain all of the letters that are enclosed in the human DNA. So let's take a step back and just talk about where DNA exists in the body. So DNA exists in every cell in the body. There are an average, at conception we start with just 1 cell, and adults have an average of about 37 trillion cells in the body, and every one of those contains a DNA molecule. Within that DNA molecule, it's chopped up into 46, is that right? 46 different chromosomes, yeah.
Yep.
So we inherit 23 from our mother and 23 from our father, and the first 22 are basically known as, we just talk about them as numbers, and then the 23rd one is known as the sex gene or the XXXY gene. That's the one that determines the sex that you're born with, and the others perform a wide variety of functions. So then each chromosome contains these long sequences of these tiny 4 chemicals, and when we write it out using those letter codes, every chromosome is millions of letters long. And researchers still don't know what all parts of the DNA do. So there's been lots of work to kind of decode it and analyze it. What we do know is that about 1% of the entire genome, 1% of human DNA, is responsible for creating proteins in the body. And proteins are basically building blocks that create tissues and organs, everything from hair to the neurons in our brains. Everything right so far, Eriskay?
Yep.
Okay.
It's good.
So we're here today to talk about 5 specific proteins, and you can see them up in the upper left. So it's GRIN1 and the 4, these are the genes, GRIN1 and the 4 different GRIN2 genes, and each of those codes a different protein. So GRIN1 codes GLUN1, GRIN2A codes GLUN2B, GRIN2A codes GLUN2A, etc. And you can see over on the right, these proteins all come together to create the NMDA receptor. And so that's sort of what it looks like. I've created a really simple version of it in the bottom left to kind of help understand in simplified terms. So basically every NMDA receptor has 4 proteins. There's always 2 proteins that are the GLUN1 proteins, and there's 2 more proteins that are GLUN2. And these can be a combination of GLUN2. So you could have 2 GLUN1 and 2 GLUN2A, or you could have 2 GLUN1 and 1 GLUN2B and 1 GLUN2C. So it all comes together. But what you need to know is that every NMDA receptor has 2 GLUN1 proteins and 2 other GLUN2 proteins. If you look at the chart up at the top, what I found interesting is that all 5 of these genes are located in different chromosomes. So they're all in different parts of the DNA. And then the final column talks about the number of bases. This is basically the number of letters that are in each set that code each different protein. So the number of letters in each gene. And so you can see from that GRIN1 is the smallest of the genes, and GRIN2B is the largest. One other interesting side note is that there's also genes in our body that are called GRIN3s, although researchers don't know exactly what they do. They called them the same thing when they were starting to name the genome because they appeared the same as GRIN1 and GRIN2 in structure, but they've since discovered that they don't appear to be part of this NMDA receptor. So don't know exactly what they do, but have a similar structure. So how do we go from this sequence of letters in a gene to creating the protein? Well, the way that I find easiest to think about it is sort of to think about Lego. So if you were to, let's say, build a toy car using Lego, you might follow the instructions, and they might say, where you put all the different colors of blocks. Well, here's a sample of just a small snippet of what could be 12 letters in that 3.2 billion letter sequence of human DNA. The way that as the DNA inside our bodies gets read is that every 3 letters creates one amino acid. So in this example of these 12 letters, these 3 letters A, C, G create the amino acid 309, I guess is how you pronounce it. TAC creates tyrosine, GGG creates glycine, and so on. So every time that there's 3 letters, they'll create a different amino acid. And all those amino acids will come together to create one protein. So if you just go, I'm just going to go back one slide, back to this number of bases, because we know that every 3 letters creates one amino acid. If you divide this number of bases by 3, you'll get the number of amino acids that are in each protein. So you can see that there's going to be more proteins in the GLUN2 proteins than there are in the GLUN1 protein. In the left here, you can see a chart that sort of shows all of the different variations that can be created. So for those 4 letters that you start with, there are 48 different words that can be spelled using these triplets, these 3-letter words. And a couple interesting things to point out. One is that sometimes different letters can spell the same word. So if you look at the very bottom right quadrant, you'll see that there's 3 different triplets, 3 different 3-letter words that all create glycine. And then if you look at the upper right, you can see that there's 3 different sets of letters there that all spell, basically tell the DNA to stop creating that protein. And so that's significant. So just remember that, because that's something that Eriskay is going to talk more about when she talks about how to read a DNA report. It's important to remember that there's also a code within the DNA that tells it basically to stop creating a protein. In the picture there, you can see 3 people with GRIN1. On the right is my son, Bryson, and the other 2 people are Olivia and Owen. And they all have the same protein change in their body. So effectively, their GRIN1 change is the same. And what's changed with all of them is the 620th amino acid. And so it should be, the 620th amino acid in a typical human should be GGG. Those triplets should be the letters GGG, which spells glycine if you look in the bottom right of this chart. But in Bryson's DNA, it's spelled AGG, which creates an amino acid called arginine. And in one of these patients, one of these 3 here, it creates that same thing. So it's also spelled AGG. But in someone else, one of the other 2, it's spelled CGG. And you can see again from this chart here that CGG also creates arginine or arginine. I'm not sure how to pronounce it. But yeah, so just interestingly that you can have a different spelling a different change in the DNA code that still creates the same new amino acid and therefore creates the same variant protein, the same changed protein. So all of these three individuals, although they can have different misspellings, they have the same protein change in their DNA. I'm not going to get into details on this slide, but this basically is a map that includes the different amino acids that come together to create proteins. And you can see for each one, that there's different triplets, different three-letter codes that create different words. But what's also different is that these all have different chemical properties. So I believe that glycine, for example, is a small amino acid that doesn't have an electrical charge. So in Bryson, where glycine is replaced with arginine, arginine is a much bigger amino acid, apparently, and also it has an electrical charge. So that's going to change the structure of the protein when it's created, and potentially also change the structure of the entire NMDA receptor when the GLUN1 protein mixes with the rest of the proteins. And before I turn it over to Eriskay for the second section, just a slide to note this is I think it's Phoenix Rising or Dark Phoenix. It's coming out this Friday, but it's a mutant movie, an X-Men movie. Just to mention that the scientists estimate that the average human has approximately 60 different mutations. And if you go back to the analogy of the car, if you're putting together a car with different Lego pieces, most of the pieces you could swap out for each other, and effectively the car might be a little different, but it would still drive and it would still work as normal. But if, let's say that instead of where the instructions told it to create a wheel, it put a square block, then that car would have a lot of challenges in doing what it needs to do. And our kids are in a sort of similar situation where, there could be all these benign variants in our body that don't really have a significant impact, but with our kids, these changes are in a very sensitive part of the body because the NMDA receptor is responsible for memory and learning, and that one change, that one letter change within 3.2 million letters can have a dramatic impact, and be very similar to that car that is missing a wheel. So just quickly to review, human DNA contains 3.2 billion letters of code. Only 4 letters are used in the DNA alphabet, A, C, G, and T. Every word in DNA is 3 letters long. And each different word tells the body to build an amino acid, at least within the genes part. The amino acids are like Lego pieces that come together to build a protein. And in every NMDA receptor, there are 4 proteins. 2 are always coded by GRIN1, and the other 2 are coded by one or more of the different GRIN2 genes. So with that, I'll hand it over to Eriskay.
Okay, great. Well, thank you very much, Keith, for inviting me to be part of this webinar. I think this is great, and I'm very happy to be here. I just want to say I'm not an expert in GRIN. I'm a genetic counselor. I've been at Sick Kids for almost 10 years now. And I've spent many years talking about genetic testing and explaining genetic test results to families, but I have not seen many GRIN families in my career so far. So I'm happy to answer, any questions that I can, and certainly happy to, receive questions that I don't have the answers to today, but would be happy to look into them and get back to people with better answers if needed. So thank you, Keith, for giving such a great primer. It sounds like you've really become quite the expert here. So I guess we'll start with the first slide. Did you advance the slide, Keith?
I did. Is this the exome sequencing slide? Do you see it yet?
Not yet. But I can start talking about it as long as everyone else can see it.
I see someone.
That's okay. So Okay. Oh, there it is. I see it now. Okay, great. So, you know, genetic testing has really come a long way. Many of you may be familiar now with exome sequencing, or you may have gone through it. But really, we've really shifted in genetics from taking a more kind of targeted approach to diagnosing genetic conditions, where we used to kind of look at one gene at a time, and you know, we did very careful assessment of the family and the patient's history to try to determine which one gene we wanted to test. And of course, this method was not ideal because, you know, there would be many genes that were, you know, that we didn't know about that were related to the clinical picture. And exome sequencing has really allowed us now to, you know, look at all of our genes in one test, and really increase our ability to make genetic diagnoses. So you can see from the picture here that exome sequencing, although it's a very powerful test and looks at all of our genes, it actually isn't looking at every single piece of genetic information. It's really just looking at what we call the exons, which we call the sort of, or used to be known as kind of the important region of the gene, or the protein coding part of the gene. So you can see the blue parts of the gene there are the exons, and the pink parts are called the introns, and that used to be called junk DNA. We used to think that that probably doesn't do much, and so what the exome sequencing is focusing on is really just the exons. So you can see nicely from the diagram of the apple below that, again, while exome sequencing covers a lot of information, it's really only just cracking the surface. We're only really looking at about 1% of our genome. In future, I think probably more and more will be using something called whole genome sequencing, which is an area that I'm working on now, where we're looking at actually all of the genetic material instead of just focusing on the exons, because you know, we realize now that the introns probably have some important information in them that are affecting how, you know, the gene is functioning. So with exome sequencing, we can get different types of results. You can get what we call a primary result, which is a result that tells us about the main reason that you came to the genetics clinic or for the genetic test, such as, you know, whether it's seizure disorders or intellectual disabilities. And then we also can get secondary information, or secondary or incidental findings, we call them. And this can be challenging in our world, because we really have to make sure and be cautious about you know, making sure that families and patients are well informed about this type of information, and well informed about their choices as to whether they want to learn this kind of information or not. But again, when you start opening up the test to look at all of our genes, we may discover more information than what we originally set out to look into. So again, I think the important piece there is to be appropriately informed about what this test can give, what type of information this test can give back to you, and whether you want that information or not. So one of the challenges in genetics is, what do we do with all this information? So, you know, when we do exome sequencing, we get lots and lots and lots of genetic variants or genetic mutations back, and it's our job to kind of figure out which of those genetic variants are actually important and potentially causing or related to the medical issues that a person might be having, and which are just, you know, benign or non-problematic variants. As Keith said, we all walk around with at least 60 different types of variants, and it's actually many more of benign variants. And so our job is to kind of sort through and look and see which variants are important. So Keith, if you can move to the next slide.
Okay.
This is kind of another way to describe sort of what Keith has mentioned previously. You know, if you look, we don't really need to go into what chromosomes and genes are, because Keith did a really nice job of doing that. But you can see this is a different way of looking at like the types of variants that we can get back. And you can see that, again, think of the DNA code as sort of sentences in a book. And so here, the sentence, the car was red, when you have a genetic change that alters that sentence so that it now reads or completely deletes one word, it doesn't make sense anymore. It's nonsensical, and therefore, using the analogy of the words being like the protein code, that is no longer readable and doesn't make sense. So you can see different types of mutations are illustrated here, and some of you may have seen or heard about a missense mutation, for example, or a deletion. So this kind of just helps to illustrate a little bit more about what exactly that is. So you can see that a missense mutation is really just a change of one DNA base pair, or one word or letter. So the car was red changes to the car was hat, or the card was RDD, which again doesn't make sense. Sometimes, though, I want to point out what this drawing or illustration doesn't show is the types of mutations or changes that would be sort of normal or wouldn't cause a problem. So, for example, if instead of was being spelt WAS, it may be spelt WAZ. The car was red. So that's obviously a spelling error and a mistake, but the meaning of that sentence is still fairly recognizable. In other words, sometimes a missense mutation can happen, but doesn't impact the actual, function of that protein. And then the insertion mutations are a little bit more, can be more detrimental because that kind of shifts everything out of frame. We can sometimes refer to that as a frame shift. Same like with the deletion mutations, where you take, one letter away and then everything shifts over. You'll see that better in the next slide. And then going to the nonsense mutations, these are also quite detrimental because what a nonsense mutation does is that it actually stops the protein from being built. So, it almost always has a very detrimental effect on the protein. Keith, if you could flip to the next slide. I'm happy also to take questions, if there are questions that come up, as we're going along.
Yeah, we have a question on chat referring to this. Could we interpret a frame shift mutation like a nonsense?
Sometimes that can happen, because sometimes when you kind of shift the whole code, ultimately what can end up happening is you reach a stop codon, which is basically like stops the production of the protein. It doesn't necessarily always happen right away. Sometimes it can happen a little bit downstream, which still can allow, some protein to be made. So it's not always the case, but sometimes, yes.
And we have another question. Would an insertion mutation be similar to a duplication?
It can be also, yes. Sometimes it depends if it's the same. Sort of sometimes duplications are sort of a certain amount of DNA that exact amount is then replicated. So that can be a form of insertion, but you don't always necessarily have that all insertions are duplications.
Okay, thank you.
Because it could just be one nucleotide that's inserted that's not actually a duplication. Can you guys still hear me?
Can still hear you.
Yeah.
I don't see any other questions right now.
Yeah.
Okay. So this is just another way to show those types of mutations, or technically now we're referring to them as variants instead of the term mutation, so we use the term variant. But a missense variant here, you can see, again, as Keith mentioned, that you have, 3 nucleotides that form an amino acid. And so they sort of go along in groups of 3, and then swapping one from an A to a C changes the protein code by changing the amino acid code. But you can also see how this sometimes has a less detrimental effect. Sometimes just changing one nucleotide doesn't necessarily change the actual amino acid. And so that might have a more minimal impact on the ultimate protein that's produced. Of course, that's not always the case, but in general, missense variants can be a bit more challenging to interpret because of this phenomenon, because of the fact that it's not as obvious what the detrimental effect is on the protein. The next slide, please.
Mm-hmm. Do you see it yet? It's changed.
Not yet on my screen, but I think this was the frame shift one.
It's a deletion one.
Oh, deletion, here we go. Yeah, so the deletion here, you can see how it causes the frame shift, because whether it's a deletion or an insertion of one nucleotide, it completely changes kind of that, again, the amino acid codes that go along with the nucleotides. And so you can see that this has a more significant effect sometimes because of the fact that it has a more downstream effect, and all of the amino acids from that point on have now changed. So hopefully that, makes it more clear to sort of understand the different types of genetic variations that exist, and how some can be more significant than others. And again, that's not a hard rule, as we know. There are many conditions that, are most often caused by missense mutations, and some others that are more often, you see more often frame shifts or stop variants in the genes. So here, Keith has very kindly offered to show the result from his son Bryson. And we can use this kind of as an example to go through and kind of talk about what a report looks like, and kind of what's important in here. What do you really need to know? And again, the lab's role is to do the analysis and do the sequencing, whether it's doing, a gene panel or one specific gene or exome sequencing, whatever it may be. Their role is to then interpret all of that data that comes back and decide what's important to report back. What does the clinician who's ordered this test really need to know? And then how they present that data is a little bit, different depending on which lab you use. But in general, they all follow sort of the same template, and we all follow, a specific set of rules and guidelines as to how to, present this information. So here you can see this is an exome sequencing report, or a snippet from an exome sequencing report. And really, ideally, I would hope that if any of you have gone through genetic testing or are going through it, that you do have a clinician or somebody who can sit down with you and really explain this to you, because that's really important that you understand what's going on in the report. So obviously the name of the condition is listed, and there's often conditions will have different names, so that can vary a little bit. But what's important here is, so inheritance pattern, so they have an AD there to signify autosomal dominant. I'm going to just briefly after this show you, you may already all be familiar with this. It may be some review for you, but if not, hopefully it will be helpful to see what that means. So it tells you how the gene is passed on, and then obviously lists the name of the gene. So I'm moving along from left to right. The position on the chromosome is not so important. This is really what's important for the lab to know. If they were going to do family testing or looking at other family members who may want to be tested for the same genetic variant, that position is quite helpform for the lab to know, but for families, it's not usually the thing that's most important for them to know. Neither is the next column, the isoform. This is, again, something that's quite important only for really the lab. The location is interesting to know. This tells you what exon. Remember my previous picture that showed you all the exons and the introns. So that shows you which exon actually the variant is found in. And then the nucleotide column is showing you the actual address or the location. So that's, you know, the actual, I refer to it as address of the variant in that particular gene and in that particular exon. And then the amino acid is listed as well, and that sort of goes back to what Keith was mentioning. You can see the amino acids the names of them are abbreviated. I think it's really small for me to see, but I think it's the G, and I wrote it down, and an R standing for glycine and arginine, and that's the position, the 620, I think it is.
That's right. Yep.
So that's just, again, yeah, that's another sort of address that sort of identifies which amino acid is changed. And then the zygosity just means, you know, is there one allele or two, or one variant or two variants? And the heterozygous means that it's just one. Homozygous would indicate that there's both, the variant is on both chromosomes. Remember, as Keith mentioned, you have, you know, everybody has 2 copies of every chromosome, one from mom, one from dad. And so in this scenario, we know that it's heterozygous, meaning just on one chromosome. And then often the lab will have their own comments, sort of like a summary of how they've interpreted this variant. The EVS is important, again, not so much, I think, probably for families or for you if you were looking at this, but it's helpful from the clinician's perspective to see. It's one of the pieces of information that tells us, or tells the clinicians or the lab, how rare this variant is. So the EVS is the Exome variant server. And so it's a large database of exome data. And so they look to see, you know, is this a variant that's seen frequently in the general population? So typically when a variant is seen, you know, more than 1% of the time, it's you know, considered what we call a polymorphism, or something that's too common to actually be really related to a health condition. So what you would want to, you know, in general, these types of variants, when we can consider them to be causing the medical condition, they're rare. You really don't see them very often. The NR just means not reported. So for now, or anyway, that variant wasn't reported at the time. The last column, I think, is an interesting one to look at. And, you know, Keith, you and I talked about this the other day. I do wonder how many people look at this column and kind of interpret maybe too much from it. So SIFT and polyphen are the names of 2 different sort of algorithm programs that are used to assess the potential pathogenicity or the sort of damaging impact that this variant might have. But it's one factor that's used to assess a variant. It's not conclusive by any means. And they often conflict with each other. They both sort of do the same thing with slightly different algorithms. And, you know, you can see in this report here, SIFT has called this variant to be damaging, and polyphen has called this variant to be probably damaging. So they're more concurrent than not, but oftentimes they disagree. You'll see SIFT says damaging and another says likely benign. So they're certainly not a reason to make a variant, you know, considered to be pathogenic or benign just by using those 2 algorithms. So that kind of explains what's in the column there. And again, I think the take-home message here is that, you know, there's a lot of information there, and I think it's important that, you know, if you're ever looking at a genetic test result, that you would consult with, you know, a clinician to kind of make sure that, you know, you have all the accurate information. And this is just kind of another way of sort of looking at the results that came from Bryson's test. I think we reviewed most of that in the previous slide.
We did, yeah.
Yeah.
So I just forwarded it now to variant interpretation.
Yeah. So again, sort of touching on a little bit further on what I said before about the process of interpretation, I think this is also really important to know is, like I said, with exome sequencing and genome sequencing, you know, we're getting lots and lots and lots of data back from these genetic tests, and really the struggle and the challenge is to sort of accurately interpret the meaning of these types of variants. We do have a sort of a body of professionals who guide us in this process called the American College of Medical Genetics. And I think what's important to know is that, you know, this process by which we categorize the variants it's based on evidence. It's an evidence-based approach, and so different types of evidence has different amount of weight to it. So, for example, a pathogenic variant, that requires a lot of strong evidence in order to actually be, you know, for a variant to actually be classified as pathogenic. So this is something where a variant has been very well described in the literature. There is multiple people with a condition that have been described with it. You know, it's a variant that is known to, or has been functionally shown to cause a problem with the protein. And so that's sort of a lot of heavy evidence that goes to calling something pathogenic. Likely pathogenic is just under that. So there's a question there. I don't know if Denise, did you catch that question?
I can read it. I've seen results on a few West reports with varying columns. What would be the reason for the information received to vary by lab? Only wondering if there's information Bryson received that's important that maybe someone else will not receive.
So, okay, I think if I understand correctly, sort of there can be variability in the reporting depending on which lab you use. And again, all labs, clinical sort of CLIA approved labs, are held to a certain standard. So you know, there should be, all of the information should be included in a report regardless of which lab you use. However, the way in which they report it can sometimes vary. And they're not always, certain labs may be less transparent about how they classified a variant. But the one that we just walked through, Bryson's report, is pretty standard. I would expect that most genetic testing facilities would produce something along those same lines. Did that answer your question? Okay, good.
Yeah.
Okay, so I don't want to get into too much nitty-gritty about these categories, but I think the important piece here is to know that, you know, there's a lot of genetic testing going on now that can often end up in a result called a variant of uncertain significance. Some of you may have received a result like that at one point. And what is that? Well, it's exactly what it sounds like. It's that they found a change, like a missense change. Often the variants of unknown significance are missense changes, you know, unless it's in a gene that's not well described to be associated with the condition. But really what that means is that we just don't have enough evidence yet to say whether this is causing the condition or not. Because like I said, it takes a lot of evidence to be really confident in calling a variant pathogenic. And the other really important piece about these categories is that they can change over time. And I think especially when you get VUSs, This is an ongoing process, as we learn more about our genes and their variants, and as we are sequencing more people, I think we will probably find more answers and more evidence that will help support either a variant being, non-causative or benign, or shifting in the other direction and potentially moving into a pathogenic classification. So again, for people who are sitting there with these variants that are either uncertain or likely pathogenic, it's really important to be in touch with your clinician, whether it's a neurologist or the geneticist or whoever it is, to make sure that over time they are checking in to reassess that variant, because again, the classification can change. I think I kind of mostly went over this in the last slide, so
okay.
I think we can kind of move forward, yeah. And then, Keith, I think this slide was really to kind of show like the different types of variants that have been described with GRIN. This isn't exhaustive, but these are ones that are from the functional database, right? And are these examples of variants that have already gone through?
So they're ones that they've either already tested or have on their list to test. So they're actual ones that they've identified. Right. But this is just to give an example, because I think, just looking through that list, probably over 95% of all the mutations are single letter missense mutations for GRIN patients, but there are examples of insertion and nonsense and deletions.
Right. Yeah. And I think, that also reiterates what I was saying, is that, you can have all different types of variants, and, each one can have a significant impact. And I think it's just, it's not that one is, necessarily more detrimental. I mean, I think the way to describe it is that in this case, you can't always attribute a specific genetic change to a specific clinical outcome or clinical picture. In time, I think as we understand probably GRIN better, we might be able to get better at doing that. But for certain conditions, that certainly exists now, where you can say, okay, if you have this variant, we know to expect, X, Y, Z. But very often, because, we have many, many, many other genes, other than just the GRIN1, there's influences from other genes, and that's going to impact how, how the clinical presentation is.
And there were questions ahead about...
And I think that's it.
There were questions ahead about mosaic. Oh, there's these ones. Do you want to talk about these and mosaicism as well?
Sorry, what was the question?
Just around mosaicism.
Mosaicism. So, mosaicism is a phenomenon where, not all the cells have the same genetic makeup. So some cells in the body may have the variant and others may not. And so it can really vary depending on, when the variant occurred. The earlier it occurred, the more likely that more cells will have it. But, sometimes it can be, can have an effect on, what tissues have the variant, and therefore, how the child or individual may be impacted. So if it's in the brain or in the heart, for example, it can have impact there, but not in other places. The one thing about mosaicism, I don't know if the question was sort of trying to get at this, but there is a phenomenon called germline mosaicism, which is where sometimes, it's quite rare, this doesn't happen very often, but sometimes an individual can carry the variant in the gonadal cells only, like in the sperm or the egg, so that the rest of their body does not have the variant, and so they appear, quote unquote healthy. They don't have the condition. But every time they have a child, they have a chance of passing it on because the variant actually exists in the gonadal cells. So, in genetics, we sometimes, when people say, oh, well, this variant is what we call de novo or new in my child, that means this will never happen again in another child. We talk about the fact that that's most likely the case, but sometimes, we do talk about the fact that there's a risk for somebody to be a mosaic carrier, a gonadal mosaic carrier. I don't know if that's the kind of answers the question or not. Hopefully it does.
Yeah, no, I think it answers. And then the other piece that you were telling me the other day is that we suspect that most of these mutations are carried either in the sperm or the egg, and so in that case, there wouldn't be mosaicism in most GRIN disorder patients.
Well, so we would suspect that the majority of these are so not carried, but that they're new in the child. So it's after the egg and the sperm are passed on, they come together right at that point when the mutation happens. And then that's the original cell that is made from the egg and the sperm. And then the mutation happens at that point, and then that cell goes on to replicate and makes all the other cells in the body carrying that variant. So yeah, in general, for GRIN, it's unusual to have mosaicism, I would say.
Do you want to talk briefly about these last two slides?
Yeah, sorry if I've taken up too much time
great.
here. Just really briefly, very briefly, this is sort of the classic picture of autosomal dominant inheritance, and kind of what I mentioned previously about Bryson's report, where it said the AD for autosomal dominant. Generally, what that means is that it's something that it doesn't necessarily mean that it's inherited like this picture shows, but what it's showing is that in an individual who has a GRIN variant, they would have one copy of their GRIN gene, which would be sort of considered, unaffected or unchanged, like the little d, and one that is. And basically, if they go on to have a child, they would have a 50-50 chance of passing that on, because they either pass on the one that's got the variant or the one that doesn't. And it's like flipping a coin each time. And so in general, you see that, as the diagram shows, that 50% of their children would have that variant and therefore have the condition, and 50% would not. And then the other form of inheritance is recessive, which I don't think you see in GRIN. Keith, correct me if I'm wrong.
I think there's one case in the literature.
Okay. So in the next slide, it shows like the type of inheritance with recessive, which is basically where both the mother and the father would have to be carriers for a condition in order for their children to have it. And the child who has a recessive condition themselves, when they go on to have children, generally, unless they're having a child with somebody who's also has, is affected or has the condition, the risk of having a child is much, much lower.
Okay, thank you. So
okay.
I'm going to go quickly through this last part so that we have time for questions. But this is just around the Emory functional analysis reports. So if you go to functionalvariants.emory.edu, that's the website for the Center for Functional Evaluation of Rare Variants. And you can see, so two things I just want to point out on this site. If you have not had your variant tested, it's something that I strongly recommend you do. And you can see along the top menu there's request analysis. That tells you how to submit your child's details to get them on the list for the functional analysis. And then the second thing there on that top menu is database, and that's where they've listed all of the variants that they've already identified. And if they've already done the testing, you can see the results of the testing. So our family went to Emory a couple of weeks ago to meet with the team that does these reports. And so basically what they do is they take a frog's egg and they basically insert DNA from one GRIN1 gene and one GRIN2 gene. So in Bryson's case, they would take his version of the variant GRIN1 gene, and they put it together with a normal, or what scientists call a wild type version of the GRIN, I think it's GRIN2B that they mix with. It's one of the GRIN2s. And then, so they have this frog's egg that has these two different versions of DNA inside it, and then they do all kinds of tests. You can see on the left the machine that they use to do these tests, but basically they test it with magnesium and with glycine and with glutamate, all of the things that typically show up or play a role in the NMDA receptor to show what's happening. And just in the bottom right there, you can see as they're tracking all these different ones, they've got some of them that they've already done the testing on, and some others where the testing's in progress. So they've got a lot that are on their list, and they're working through them. And this is Bryson's report. So the actual report is maybe 3 or 4 pages long, but this is the most important piece if you get your report back. Basically, it's the summary of results. So you can see glutamate, glycine, magnesium along the left, some of the key things that they test. And basically, so it's a one regular. So the first column shows what happens if you take a regular GLUN1 protein and regular GLUN2 protein, and then the second column is if you take the regular GRIN2A protein and mix it with the GLUN variant that Bryson has, and you can see how it reacts differently to all of these different chemicals. And the very last column is basically what they do is they take it and they do the math to kind of show if it seems to be having an effect of loss of function or gain of function on this kind of mini 2-protein NMDA receptor in the frog's egg. And so, in the case of Bryson, it's interesting that most of these tests suggest a slight loss of function. So wherever you see the minus before a number, that suggests loss of function. But the biggest number is around magnesium, where there's a 19-fold change in magnesium, which basically what the hypothesis at this point is that because the NMDA receptor kind of opens and closes based on electrical charge, when it closes, it does so because the electrical charge makes it fill up with magnesium. And so the theory with Bryson and the 4 other patients who have the same protein change is that because there's all this magnesium, or it's the opposite. There's not enough magnesium going there. Basically, the NMDA receptor in these kids is always open. So that's the theory. We've got a mouse model that's being created with this, and we're also, part of what we did at Emory is also getting what they call mini brains done, and so that they can actually test better and get a better understanding through the mouse model and through the mini brains to find out if it backs up this hypothesis that magnesium is the factor and understand what's going on in Bryson's brain. And then here's just 2 other screen grabs that I took from the Emory site where they've got the functional analysis summary. But you can see here there's a bunch of GRIN1 changes, and almost every one of these, I think every number here is a positive, so there's no minus sign. So that means that pretty much all of these variants that they classified, their hypothesis is that these are all gain of function. And just looking at the report, it seems that there's certain regions of the NMDA receptor where gain of function is more likely and certain where loss of function is more likely. And here's one where, this is GRIN2A. The very top one is mixed results. Some of the others show gain of function. And then there's this one with the funny symbols, the 5th one down, where it's funny symbols all the way across. In this case, basically, from what I understand, the electrical current isn't reaching the outside, so they actually can't complete the full test on some of these variants. So there's a question here.
Hey, yeah. So a question from Zoe, if it's always open, is that gain of function?
That's the theory. So, I mean, I think with all of the early, and I mean, from some scientists that I talk to, I know Amy Ramsey here in Toronto often makes this point that we have to be really careful about making assumptions that are too broad based on these functional analyses. So, you know, I think of it as maybe something better than a best guess, but not conclusive. And the variants without numbers are the ones that are, have not been tested yet. So they're on the list to test, but they haven't got to yet.
Thank you.
But it's interesting just going through there and looking at all of the testing that they've already done. And I know that there's labs in Europe that are doing similar testing, but I can't speak to that. I don't know if they do them, use the same testing model. Barcelona, Zoe says. So that's kind of what we wanted to go through. And if you have any questions, you can either type them in the chat or you can take yourself off mute and just ask away.
Are there any more questions? You can unmute yourself on the participant list or ask in chat. What do we have to remove to identify our child to submit for analysis? I think she meant, what do we have to do?
So I don't know I don't know specifically. What I know is that they do the functional analysis just based on you know, you telling them the protein change. They don't need the blood. They just, because that's already been done, so they can just build it. They can just build it from what you provide. So I don't know if otherwise. I don't know if there's you know, you're not publicly listed, but I don't know if how they. I don't know what, I can't remember what the details are of submitting and identifying.
And another question.
I thought.
Go ahead.
No, go ahead.
We have a question. Just very basic. What is functional analysis, and what does the word function mean in this case? It's a good question.
Yeah. So as far as I understand it, it's basically trying to it's tests that they do to try and understand what's going on in an NMDA receptor as a result of the variant in the gene. So, you know, as we talked about before, a typical NMDA receptor has 4 different proteins. They basically try to create a mini NMDA receptor with 2 proteins, and then basically try to figure out gain of function versus loss of function. So, and that leads to another question I see here: loss of function or gain of function. Basically, the way that I've heard it explained is that you know, the NMDA receptors are in our brains, and it's how we learn and how we develop memories. And basically, there's a conversation going on in our brains between different neurons. And if there's a loss of function in the NMDA receptor is basically whispering. If there's a gain of function, it's basically yelling. And neither of those is good. You want your neurons to be able to communicate at kind of a level, not too quiet and not too loud, right? Just like Goldilocks and the Three Bears, you want it to be just right. And so, so that's, you know, so with all of the GRIN patients, they either have gain of function or loss of function, but neither has a good outcome. Both of them have an outcome where it ends up, you know, whether the NMDA receptor is underperforming or working on overdrive, it has implications and can create, you know, with some kids seizures, with some kids, most kids learning disabilities, low muscle tone, different things depending on the gene and depending on the specific protein change.
Keith, can I just add to that?
Please.
It's pretty important to find this out, because depending on whether you have gain of function or loss of function, that can have an impact on what kind of drugs are suitable for your child as potential treatment. So this is just a kind of a heads up to anybody who hasn't had their functional analysis done. It is pretty important. It gives scientists an awful lot of information, but also it gives you the potentials to try and find the right therapy, the right treatment for your child.
Yeah, that's a really good point. And, we're at a point now where there's not a lot of great treatment options that we've found so far that are making dramatic differences in kids with either gain of function or loss of function. But I think we're at a point, where researchers and biotech and pharma companies are starting to spend more time with this, and we may, be at a point before too long where we have better therapies, but we'll need to know, functional analysis and what's going on before we can use those. Any other questions? Denise, do you want to wrap it up?
Okay, well, thank you everybody for joining us. And like I said, this is recorded. We will be posting the link to our groups as well as YouTube, and we'll let you know when that's available. And Keith, thank you so much for leading this, and Eriskay, thank you so much for joining us as well.
Thanks, everyone.
Thank you.
Thank you. Bye-bye.
Bye.
Bye-bye.
Thanks so much, Keith. Bye.
Bye.